So, good afternoon and welcome to the presentation here of FluoGuide. We are so eager to present it for you as we are two people, so both Andreas and myself will present it. So what we are doing is that we improve the outcome of surgery, and we do that by intelligent surgical targeting. First a slide just saying that the future is difficult to predict. It's also difficult for us. What we are starting out with is in cancer. And there's about 15 million patients that have cancer. And the problem is that many of the cancer will reoccur locally after surgery. So after the surgery, about 50% on average will have cancer reoccur locally. And of course, that's not the whole idea with it. And as you can see, it's different from the different indications. Glioblastoma, for instance, is almost 100%, whereas on other cancers, colorectal cancer is less and about 20%. That even 20% is quite a lot if you imagine it. So what we are doing, we have an UPAR-targeted fluorophore that light up the cancer so the surgeon can see it and remove everything. This is a novel supportive technology, and what we mean with this is that actually it's a technology where we help big players in the field. It's the surgeons, it's the equipment manufacturing, and even if we are small in size, we can have a huge impact in the whole benefit of the clinical trials. We're an experienced team. We're focusing in on glioblastoma, but our market potential is going far beyond glioblastoma, and then we are listed here on Spotlight. Surgery is a cornerstone in early cancer treatment. The surgeon want to remove the cancer precisely, but spare normal tissue. And it's obvious because the patient increase the life of survival, chance of survival. The hospital reduce the spend of resources, and also the surgeon is benefited from it. The surgeon is doing a lot of important decisions when doing the surgery. They look into the field, and the first cancer can be easy to remove, but then it becomes more difficult because there may be something very close to a vesicle or a nerve, and should the surgeon take it out with the risk of the patient having disability afterwards, or should they leave it with the risk that the patient have reoccur of cancer? So we help them because it takes time to do this surgery precisely. So we help them, and of course, precision and speed obviously benefit from all of them. And as you can see, there's many different kinds of surgeries. There's laparoscopic surgery, microscopic surgery, open surgery, but they all benefit from increased precision. We have a small film here, 1.5 minutes. If you know FluoGuide, you will know the movie, but if you don't, it's a good introduction to it. So it's coming here.
When performing surgery to cure patients from cancer, it's difficult for surgeons to distinguish the cancer from healthy tissue. In approximately 50% of all cases, there are actually cancer cells left behind, risking the cancer to grow again and spread. FluoGuide develops a substance discovered by Danish researchers, FG1, a fluorophore that binds to cancer cells and makes them light up. It is administered to the patient just prior to surgery. The new technique utilizes the fact that cancer cells express a specific receptor, UPAR, which FG1 binds to, making the tumor fluorescent, thereby assisting the surgeon to remove all cancer without cutting out more healthy tissue than needed. This is what we refer to as intelligent surgical targeting. It can give thousands of cancer patients a better outlook without increasing the cost of care, as this new technique will mean enormous savings on reoperations and other treatments. So far, preclinical studies have shown that in half of the cases, our technology could find cancer left behind after standard surgery. Now, the next step is to test FluoGuide's first product in patients with brain cancer, which starts with production of FG1 and safety studies. We are also developing FG1 to help patients with cancer in locations other than the brain. With a clear path to commercialization and support from all stakeholders, we believe the introduction of this technology will be quick, as the benefits of FG1 are quite clear.
So, hope this gave an introduction to our core technology. I'll take you a little bit through some of the details and where we stand now and what we have shown. As the movie showed a little bit quickly, it's a very simple technique. You give an injection prior to surgery. The patient, when anesthesia is induced, there's an IV line. Anyhow, our product FG1 is injected. It then binds to the cancer and gets excreted from everywhere else, so you have the cancer then light up, typically after 30 minutes, and that will last for a long time window. And then when the surgeon starts the operation, you can use a special light, a night vision if you want, and this will make the cancer light up. So it's easy to see what is cancer or not. So it has a lot of advantages. It will be a one-stop shop, so you don't have to go back. If you have left some of the cancer, you can do it better. The chances of survival thereby increases. If you leave behind cancer cells, the prognosis is known to be very poor. It's cost-effective because you save on the reoperation and you do everything better in the first run. And it's less stress for the patient, one stop, and it increases speed and quality and how sure the surgeon is on what the surgeon is doing. The target we are approaching, UPAR, without becoming too technically, it has all the properties it should have to be perfect for this use. It's highly expressed on cancer cells in most of solid tumors actually, and it's not really expressed in the background. So you have a high tumor to background ratio, so only the cancer lights up. And then there are several hundred papers linking UPAR to cancer, and we know very much about this system. And we have proof of concept in humans. On the right, you can actually see this is a brain tumor. The whole blue part is the brain taking out, and what is darker blue is the tumor. And you can see when you then put our technology on, it lights up in green. And you can also see there's a small island you might have overlooked on the right hand. So we have been performing experiments so far in animals. Morten will come back to when we move into patients in the not too far future. But basically what is done is before surgery, we inject this compound, it binds, and then we do the surgery with a surgical robot. And you can actually see in the middle how it looks in white light. This is what the surgeon sees. The surgeon has to find out what is tumor, what is not tumor. And then when the night vision is turned on and we injected FG1 half an hour earlier, you get the next image, the green one. And then suddenly it's very easy to see what is cancer or not. And if you move to the last image on the far hand right, you can actually see then you do your surgery, you remove what you think is the cancer, and then you turn on the light again and say, no, there are some small green parts left, and that is actually what you then can take out. We do this in mice, but when you do mice experiments, you actually do human cancer because you implant human cancer into a mouse. And we did recently a study that has just been published, what we call a mouse clinical trial. And why do we call it a mouse clinical trial? That is because we mimicked everything just as we want to do it with the patients soon and did that in mice instead. So we injected this compound in mice that had human pancreatic cancer, implanted a very severe cancer, you heard that previously. Then the surgeon did the operation, and this is real surgeons that are used to do human surgery, removed the tumor in white light. And then they said, I'm done, and then we turned on this special night vision. And in half of the cases, we could say, you didn't do your job fully yet. There's something left. So half of the mice, we found extra cancer tissue. And if you leave cancer tissue behind, you're not going to be healed. So very large expectation when moving into humans.
Okay, thank you. So now, as Andreas said, it's prepared for clinical trials. And as I said, it's been proven in different cancers, glioblastoma, head and neck, and pancreatic cancer. We have been working heavily the last half a year on setting up the production and the toxicity testing, which is needed for us to go into man. That's to do it properly. We have had established the synthesis method before, but now it have to be put into vendors that are specialized to do it in a quality that we can use for clinical trials. So we have now the supply chain established so we can supply to clinical trials. We could do the formulations, and actually we have we are ahead of plan here that we almost have a commercial synthesis set up, which we're quite pleased with because it's a fluorophore, it's light sensitive, it's not that stable. So having a process where we have this under control is one of the things we consider as a critical thing to get into man. So we're very pleased that we are ahead of schedule here. It's a high likelihood that we will be well tolerated in man, as we have 2 building blocks in the product, both being tested in man, and that has been well tolerated. On the clinical, on the toxicity studies, what we're doing right now is that we have to qualify the species we are testing it in, because different species, different animals have different UPAs, and we have to show that our product is binding to the UPA in the animal. So we are screening all the animals through from rat to monkey, and there will be 2 outcomes of that. Either we'll have to do the study in monkey, and then we have a third quarter result of our first clinical trials next year, or it will not be done in monkey, and we have it in second quarter next year. So that's the 2 outcomes of it. All the slots are booked, and we're ready to go. And it's like it is. On glioblastoma, it's a huge unmet medical need, and if you look at the 5-year survival, 1 out of 20 patients will survive out of 5 years only. So it's a very severe indication, and almost all of the cancer recur locally after surgery. So it's very relevant to use a technology like ours. 16,000 patients per year has it in Europe and US, and that's qualified for an orphan drug designation. So the interesting thing with FluoGuide is that our development, our product is a medical product, so it's a pharmaceutical product under the law of pharmaceuticals. It's an imaging agent, but the interesting thing is that we move very quickly from where we are now until commercial stage. And one of the reasons for this is that the clinical trials is very simple and very straightforward. So it's you have to include the patient before surgery, exactly like the mice clinical study, as Andreas explained before. You include the patient, you do the surgery under white light, switch on the fluorescent light, and you have a result. So the study is very quickly. Another thing is that there's no placebo control because the patient are their own control. So we don't need a huge portion of patients to make a control group. We don't need any randomization. And last but not least, the competition of us going in for the cancer patient, I know it can sound a bit crude, but it's actually an issue when developing cancer product. That's a lot of medication that is being developed, and it can be difficult to recruit the patient. We have no competition here. Every surgical procedure that is done can use our product on top of it, so all patients can be included. And we need 2 studies to have an approval. The first study is being initiated next first half next year, and we have the result in second or third quarter coming year, and that's a 10 to 20 patient study in glioblastoma. The next study then will be signed exactly similar. It's a 150 patient study, and that will lead to approval, assuming an orphan drug designation for glioblastoma. So an interesting UPA is not only very extensively expressed in glioblastoma. So we have 3 of the big, largest indications. So that is the breast, colorectal, and lung cancer. UPA is also extensively expressed in those indications. And it means that our product can cure or can help curing those patients as well. In the surgical procedure, everyone benefits from the product. FG1 is injected prior to surgery. The surgery takes place, and then just right after surgery and following surgery, the patient are looked for relapse or if the cancer recur. And if they have, they go back into the procedure and have either a new surgical procedure or more aggressive treatment. But interestingly, we increase the efficiency in the operation. We less stress to the patient, decrease the cost. And last thing also is very important that we help the manufacturers of equipment that they can get better value out of their equipment. So the equipment manufacturers are quite pleased to have something like our product that can help them guiding the surgery. Our customers are very high valuable, and there's not that many of them in the sense that they're very concentrated. So it's the surgeons around the world, and there's 1 million approximately surgeons around the world, divided on 12,000 hospitals. We already start now interacting with them through the clinical trials preparation we are doing, and get good feedback on the indications. Also, it's important that there's not just one partner that can help us or take on the product further on. There's all the big medtech companies are available in the field, the pharma companies, imaging companies. So there's a range of partners that can either take it for area, take it completely, or can help us in the distribution. And very importantly is that the first group we are going out to, the neurosurgeon, there's only 50,000 neurosurgeons in the world. So it's a very small group of customers we have to go to start with. In terms of competition, that was started out in the beginning as a down right corner. There's more mechanical equipment that could, for instance, ultrasound, that could see the tissue. It can be used on all cancers, but it's not very specific to the cancer. Then moving to the left, there are, for instance, 5 ALA for the glioblastoma. That was the first generation product that could light up cancer. It's not very specific, and it does not work for many indications. It's not very specific because it's converted in the cancer and form a cloud around the cancer. And that means when the neurosurgeons see this cloud, they say, okay, well, I take out more than actually they need. And what happens is that the disability of the patient in meta-analyzed study that is being meta-analyzed have a higher disability when using 5 ALA. And the neurosurgeon is kind of 50-50 interesting in this product. But the good thing is that there's a product out there that start building the technology that you should have something to guide the surgery. Then there are the range of, first generation active targeted FluoPhore that binds to the cancer cells themselves. And they are very specific, so that's, for instance, the CEA is a target for colorectal cancer. It can only be working colorectal cancer, but very specific to it. Or there's an untargeted company that have the B vitamin receptor. It works very many places, but also normal tissue, meaning that a lot of normal tissue is removed. So it's a kind of a balance. So we are the only one that have a very specific target to cancer, and that is very broadly, that goes for all the cancer patients, or most of the cancer patients. And interesting as well is that the untargeted therapeutics are now in phase 3. So they kind of paved the way through the system, the regulatory system, have the discussion with regulatory agencies, and now we can follow after them by learning from the studies they are doing. So we're very well positioned for that. One question we often get is, how big is the market? There's no market yet for active FluoPhores out there. So just making the list of different elements that contribute to the market, and then you can do your own math. We will hesitate to put a number on it, but it's very important that you have the cost of the surgery, you have the relapse of the patient that get local relapse. For instance, on glioblastoma is 100%, breast it's only 30%. You have additional costs for care, because it's more expensive if you have a patient that is not completed operated. You have the cost for disability you get with unprecise surgery. You have the stress, and that's quite a lot, because if you think about breast cancer, for instance, you don't get disability but not removing it correctly. But actually, it is very important for the females that do not have a complete removal of the breast cancer. So it's a very valuable issue that you reduce that. Then you also have the saving because of the surgical procedures, and of course a number of patients. So there's no doubt that this is a multi-billion-dollar market, and it's not so much a question if the market is big. It's more a question how to get in there and behave in a optimal way, and that's what we work hard on. We have a very strong protection. We have patent protection on it. The first patent has been issued now in the US, and we have a range of applications that comes in over the next period of time. On an organization is Andreas and myself, and then Grete is taking care of the production and the early toxicity. She's done that before. It's quite important. It's a discipline that is very specialized, and she's done that before, brought product to the market, done the production, done the tox, done the early clinical trials, and that's a kind of a discipline in herself. And then we have a very good board with the international. We have one from Sweden, we have one from US, West Coast, tech investor. We have worked earlier in Medtronic, and then we also have Arne, that is our chairman, been in several Swedish companies as well. So our plan was that we went out raising 20 million Danish crown, including the one that we issued here in the springtime. And that would bring us a proof of concept for one indication for one product in glioblastoma mid of next year. We have prepared the partnering, so we've already done that. I can say our presentation will be available on our homepage, so you don't need to take photos. We'll send them out. We have prepared the partnering. We start already now on that, and we expect that we can start compassionate use sale end of next year based on our first clinical study, and as well doing a partnering agreement. We have a very strong news flow. We have a commercial supply chain will be set up. We have the result of our first regulatory meeting. We have a submission of the clinical trial application, the result of it. We have the warrant issue. We have commercialization in terms of patient compassionate use and a partnering, and then the warrant issue. And just last, we are traded on a spotlight. If you're interested, we're also around if you have any questions.
Tack för det. Spännande. Mycket på gång. Vem vill öppna upp? Vem känner sig manad att ställa första frågan? Ser jag någon? Hej, då passar jag på. Du pratade lite om competition. Vad är det för competition där ute? Finns det något där gör samma sak eller nästan samma sak där gör?
Yeah. There are the companies that work on the active fluorophores. I will mention the Untitled Laboratories is a company that is most advanced. They're working with this B-vitamin receptor target, and they work for ovarian cancer. So we believe that more competitors in the field that can push it forward would be nice. They're not direct competitors in the sense that they work on ovarian cancer and vitamin B receptor, but they help us moving up the field. And of course, this will be a big market in the end, and there's no doubt they will move into other indication already. They're working on it, and as well will we. So I don't believe that any one company will own the whole space. We will kind of divide it with different indication, different products, most likely, but even that is very attractive.
Okej, tack för det. Någon som vill passa på? Kommer.
Can you elaborate a little bit on the business model, how you will take this to the market, and will it be with partners or etc.?
Yes, of course. I mean, our plan is that the sweet spot for doing the first deal will be after we have the clinical result. That means after the result mid of next year. So that's why we expect that we will have a partnering deal end of next year. What we will aim at is that we'll aim of a regional deal where we will sell out Asia, China, because that's very complicated. It's a very big market. It will be the second biggest market in the world, but it's also a complicated market, so it makes sense. And part of that proceed we will then use to further develop it in Europe and in US. As mentioned, that we have a very limited customer group, so we can start the pre-sale. We can do that ourselves, and then we're in a very good position to make a larger deal at that time.
Okej, tack för det svaret, Morten. Ja, ett ögonblick.
Jo, ibland finns det ju enstaka cancerceller kvar. Vad är den minsta ansamling av cancerceller som lyser upp så att en vanlig människa, operatören, kan se det där som fluorescerande ljus?
Ja. This is a very good question about how many cancer cells can be seen, and the easy answer is it cannot see one cell. So it's kind of 50 cells or 100 cells as the technology is right now. For neurosurgeons, probably less, because they do their operation routinely on a microscope system. So there it's probably very few cells. You will probably leave back single cells, but what we know, we studied very in depth how our fluorophore gets to the single cells, because in brain tumor it's characterized by these seeds around, and that's actually why 100% get relapse. It seems to be in certain areas that there are many of these islands, and that will as a whole certainly light up at a lower level, but that can be seen in the microscope. The single cells that might be back, you still have to remember that it would still be combined as a routine with radiotherapy and chemotherapy. So there's also a threshold where you will be able to heal the patient if you get below that. And exactly that question we have looked into. So we expect that we will be able to remove exactly that extra amount of these single cells or the areas with islands that will make it possible to heal the patient.
Okej, tack för det svaret. Tack för att ni kom hit.
Ja, tack.
Båda
Tack för
två.
det.